Last week, we started with naming of enzymes and classification of enzymes. So we did see how enzymes were named, looking at different systems of naming and classification as well. We looked at different systems of classification of enzymes. We are continuing today to look at what we refer to as coenzymes and cofactors. So if you take an enzyme, the last time we got to know that enzymes were mostly proteins. But of course, we also got to know that some enzymes were some catalytic RNAs, which were mostly involved in the processing of nucleic acids themselves. So if generally you have these enzymes being these biomolecules are either proteins or nucleic acids. Generally, these proteins are inactive without a non-protein part, what we do refer to as the coenzymes and the cofactors. When you read biochemistry, a lot of times these two terms, coenzymes and cofactors, are used interchangeably, but in some instances also you find that they are actually distinguished. Yes, there's a difference between what we refer to as coenzymes and what we do refer to as cofactors. In some cases, all of them are referred to as cofactors, but not all cofactors are coenzymes. You'll find some of these terms being defined that way, so that yes, all non-protein parts which are required for the activity of an enzyme it can be referred to as a cofactors, but not all of them are coenzymes. And we'll come to see the difference between these two terms, the coenzymes and cofactors, as we go on. But here, what we have on our screen is a depiction of what we do refer to as an apoenzyme. Sometimes you also see it being just called apoprotein. And that is the protein part of the enzyme, the inactive part of the enzyme, which requires the cofactor or the coenzyme to bind to it in order to make it active. So that coenzyme or the cofactor is an activator. So you do remember one of the terms that we studied in our last lecture was activation and activator. Now, it's just like how I think when we were looking at the structure of proteins, we did talk about the structure of hemoglobin, for instance, that was having a protein part, and then there was a ion, Fe2 plus, which was at the center of this hemoglobin molecule. And without that ion, that hemoglobin will not be able to carry out its function of transportation of oxygen from the lungs to the tissues, and carbon dioxide from the tissues back to the lungs to be exhaled out of the body. So here too, yes, the enzyme requires this non-protein part for it to be active. And when you have the non-protein part, which is that apoenzyme or the apoprotein, which binds to the coenzyme or the cofactor, the non-protein part, together, that complex is what is being referred to as a holoenzyme. So holoenzyme, same as say the active enzyme as a whole enzyme. So here you see that the enzyme is depicted to be having an active site that has a configuration which is not fitting to the substrate, and that a coenzyme, after it binds into that active center of the APO enzyme, it makes or it changes the configuration of the active site of the enzyme so that it is now fitting to the substrate. So now it can interact with the substrate and catalyze the reaction on that the reaction that substrate it will be undergoing. So you have learned some at least two other terms today which we didn't see the last time. Or I think even APO enzyme, I think we saw it. Maybe it's only one hollow enzyme that we didn't see the last time. All these other terms, I think you have seen them. The coenzyme cofactor, we did talk about it. Activator, we did talk about it. Substrates, yes, we talked about it. So the new term which you can pick from this is hollow enzyme, which is the active enzyme, the whole complex coming from the enzyme, which is that protein part, and the non-protein part, what we are referring to as a cofactor or the coenzyme. Now we want to distinguish between these cofactors and these coenzymes. Usually, when you hear the term cofactors, this refers to inorganic metal ions, like as you've seen here, some copper ions, ferrous ion, ferric ion, potassium ion, magnesium ion, manganese ion, molybdenum, nickel ion, selenium, zinc. These are some common cofactors that you find binding to the apoprotein to activate them, making them active to carry out their enzymatic reactions. Now, what it means is that you know that a protein, yes, a protein is a sequence of amino acids bound together. You can have a large protein. You can have maybe some even enzyme can be dipeptides, tripeptides, tetrapeptides, oligopeptides. You have polypeptides, really big proteins that consist of hundreds, thousands of amino acids. You have these cofactors that are actually required to when they bind to some specific portions, particularly the active sites of these enzymes, they give these enzymes a particular configuration so that they are able to interact with their substrates. So every enzyme will have a certain cofactor that it requires to be active. So we are seeing some examples here that you have copper ions being cofactors to some enzymes that we refer to as cytochrome oxidases. You'll find cytochrome oxidases in the liver. They are involved in what we refer to as biotransformation. That is just like you do have digestion of food. When you take in food, the food goes through the digestive system. You remember all that process of digestion. Similarly, when you take in some chemical substances, it could be some medication, or maybe if by accident some toxic substance gets into the body, the liver is responsible for transforming those substances to be excreted out of the body. So the liver carries out what we refer to as detoxification. But of course, it's not all the reactions that the liver, carries out that becomes detoxification. In some cases, even though what is intended is for the substance to become less toxic and get excreted out of the body, sometimes a substance could actually become more toxic, what we do refer to as toxification. But here, what we are talking about is that, the say cytochrome oxidases, this you'll find, say, in the liver, they require some of these metal ions like copper ions, ferric ion, ferrous ion, enzymes like catalase, peroxidase, all of these require these ions to be active. Potassium ions for, say, pyruvate kinase. We did talk about what kinases were the last time. I did mention that kinases were enzymes that were involved in the transfer of a phosphate group between one molecule and the other. Usually you have phosphate groups that can be transferred from, say, cellular energy, ATP, onto a molecule as a form of activating that molecule. So that's a process of phosphorylation. You find potassium ions are cofactors to an enzyme such as the pyruvate kinase. If you did metabolism of, say, for instance, carbohydrates, you would have come across, say, pyruvate kinase, which was responsible for the phosphorylation of, say, pyruvate, for instance, which is the end product of the process of glycolysis. Before that, pyruvate, undergoes process of decarboxylation and goes into the Krebs cycle. Magnesium ions also are cofactors to hexokinase. I think we also saw hexokinase the last time, glucose 6-phosphatase. So now clearly, when you see the names of the enzymes, you will be able to just relate to them to say, okay, glucose 6-phosphatase, what will that enzyme be doing? That enzyme will be hydrolyzing phosphate group from the glucose 6-phosphate. Pyruvate kinase, we have just talked about it. Manganese ions can also be cofactors to arginase, ribonucleotide reductase. So you do remember the amino acid arginine. So that will be an enzyme that will be involved in the breakdown of arginine. Ribonucleotide reductase will be involved in reduction of the ribonucleotide. Or you can say it's an enzyme that acts in nucleic acid metabolism. Molybdenum dinitrogenase, nickel ion being a cofactor to urease. We did see urease, an enzyme which was involved in the hydrolysis of urea. Selenium, glutathione peroxidase. So that will be a cofactor to the oxidation of glutathione. Carbonic anhydrase, zinc ion, there will be the cofactor. I think we did see the conversion of, say, carbon dioxide to carbonic acid, and then from there producing bicarbonate ions. I think when we're talking about mechanism of like last semester when we were looking at proteins, and we saw a bit of functions of proteins. I think we did talk about this, but well, even if we didn't talk about it, you can read just a little bit about what carbonic anhydrase is, and you will understand it. Alcohol dehydrogenase. I think in this lecture we'll see an example of a reaction that alcohol dehydrogenase is involved in. Carboxypeptidases. These are also, enzymes involved in the decarboxylation of proteins, peptides, or let's say amino acids. So specifically, when you see cofactors, this is what they are referring to. inorganic metal ions, which are required as activators of enzymes. Then what are coenzymes? What then are coenzymes? So the coenzymes, these are not metal ions. These are organic molecules. These are usually, and they are derivatives of, you know what we do refer to as vitamin B complex. You have different vitamin Bs. And so you see on this table, what we do consume as vitamins, which serve as dietary precursors for these coenzymes. So for instance, what we do consume as a vitamin, biotin. I think biotin is vitamin B7. That vitamin B7, take it out, it is in its inactive form, and it gets activated into a coenzyme, what is being referred to as biocytin. And what does biocytin do? Biocytin is a coenzyme which is involved in the transfer of carboxylic groups, say in the form of it's written as CO2, not necessarily the carbon dioxide gas that we refer to, but in, say, the process of decarboxylation, where carboxylic acid group is removed from, say, carboxylic acid and being transferred onto another, you will see biocytin is acting there as a coenzyme. We'll see examples of some of these reactions so you understand it better. And we also see another coenzyme, pantothenic acid, is also another form of vitamin B. I don't really remember. I think it's maybe vitamin B5 or B3 or so, and that's actually is what we do call coenzyme A. We will see the structure of coenzyme A. I think for now you have seen coenzyme A being mentioned in many biochemical reactions already, but perhaps you haven't seen the structure of the coenzyme A yet, because every time you see it being written as a CoA. But today we'll see the structure of the coenzyme A, how it looks like. But you just have pantothenic acid being part of that molecule because the molecule is much bigger than pantothenic acid. It's also involved in the transfer of acyl groups. So if you say an acyl group, it only means that, for instance, you have methane, and if you have methyl group, the methyl group, that is CH3. If you have an ethyl group, that's a CH2, CH3. These will be called the alkyl group. But if you have, say, acetic group, that's an acetyl group from acetic acid. You do have some derivatives of that, what you like coming from fatty acids. When you say an acyl group, that is coming from, say, a fatty acid, say a fragment of a fatty acid. There's a sort of, let's say if you take palmitic acid, and it's broken down, and you still have the carboxylic acid group there with some carbon chain on it. That's an acyl group. You have vitamin B12, which is involved in the transfer of protons. Though it's referred to as hydrogen atoms here, but sometimes you see it as being transfer of protons because, of course, a hydrogen atom is usually that's an atom with a proton and an electron. So it's usually a transfer of one electron, one proton. Also alkyl groups coming from, say, methyl, ethyl groups. Its activated form, you see, it's deoxyadenosine cobalamin. So there you see that there is also a metal in that structure, cobalt. And you also can see that there is a deoxyribose sugar in there, almost like a derivative of the nucleotide deoxyribose in nucleic acids, as you would have seen, because I'm sure by now you have done some things on DNA and RNA. And you see riboflavin, vitamin B2, involved in the transfer of electrons. That serves as and is in its active form is what you see usually in biochemical reactions as FAD. That is flavin adenine dinucleotide. We'll see the structure as we continue. We'll see the structure, and we'll also see the functions that this carries out. And we have some other enzymes, the coenzyme lipoids. These are not from any diets. They will usually, like as from the name, they can get synthesized by the body itself. The name will tell you there are some derivatives of some lipids. Nicotinamide adenine dinucleotide coming from niacin. I think niacin is vitamin B3 or so. That will be involved in transfer of some hydrides. Pyridoxal phosphate that's a coenzyme that you see in protein metabolism. In the transfer of amino groups, a process that we do refer to as transamination. You see pyridoxal phosphate. It's coming from the vitamin B6. Tetrahydrofolate, what you usually hear as folic acid, is coming from that, folate. If you say folate is the same as folic acid. It's involved in a transfer of one carbon groups. We'll see these things much more, each of almost every single one of these ones, even not everything, but most of it we'll see. We'll take a look at their structures and the functions that they carry out. And then the last one, the vitamin, the vitamin B1, which is thiamine. From it, you have the coenzyme thiamine pyrophosphate, and it's involved in metabolism of aldehydes. So we'll see these things one after the other and how they carry out their reactions. From this table, you will not understand it fully, but if you look at the structures and the various reactions, you will understand it better. So we take the first one, which is thiamine pyrophosphate. But even before then, I want to draw your attention to the fact that you could now see that a lot of these enzymes, or this what we are referring to as a coenzymes, you will see that we are referring to vitamin B, different vitamin Bs, different ones that are acting in their active forms as coenzymes. And I don't know if I mentioned it to you the last time that many of these vitamin Bs, because they are involved in these biochemical reactions as coenzymes, you realize that when medicines are being prescribed a lot of times, you see vitamin B complex being prescribed in addition to these medicines. And that's because these vitamin Bs are required as coenzymes, which will aid or activate the enzymes which are involved in the metabolism of the drugs that are being given to whichever patient. So these are very important components, the vitamin Bs involved in the what we call drug metabolism. So we take the first one coming from vitamin B1, which is thymine pyrophosphate. Now in biochemistry, when you see pyrophosphate, it is the same as diphosphate, meaning two phosphate groups. So this is vitamin B1. On the right-hand side, you see vitamin B1. That's thymine. If you look at the structure and compare it to the structure which is on the left-hand side, what we are calling thymine pyrophosphate, and I just mentioned that pyrophosphate means diphosphate. Sometimes you see biphosphate. It only means two phosphate groups. So we can see clearly two phosphate groups on that molecule, the thymine pyrophosphate. All the other parts of the molecule is actually coming from thymine. It's the same thing. This the six-sided ring is the same six-sided ring you see. That CH2 you see there is the same as that. And the five-sided ring, the thiazolone ring, is the same thing which you see in thymine pyrophosphate. And the end, CH2, CH2, there too you see, even though it's not being written, you see there will be two CH2 there. And in place of the two phosphate groups, you have a hydroxyl group. So thymine, the vitamin which we consume, is in its inactive form, but it gets activated by phosphorylation. That is two phosphate groups coming in place of that hydroxyl group makes the molecule active as a coenzyme, what we are referring to as a thymine pyrophosphate. Now, the very active portion of this molecule is the thiazolone ring, and the particular portion of the thiazolone ring which will be involved in the catalysis of the chemical reactions is this hydrogen on this carbon number 2. So that is why intentionally we've made it red. So you can see that is the reactive portion of the molecule. So the thiazolone ring, inside the thiazolone ring, you have carbon 2, and on carbon 2, there's a hydrogen. And it is that particular portion, it is on this carbon 2 that you will be having the reactions that the coenzyme catalyzes, the coenzyme interaction with the substrate is actually will be occurring on this carbon number 2 of the thiazolone ring. We'll see the mechanism of action of this coenzyme, and so you will understand it much better what we are talking about. Now, what are the reactions that the thymine pyrophosphate is involved in? So we have thymine pyrophosphate being a coenzyme to pyruvate decarboxylase. So these are some examples of enzymes that thymine pyrophosphate is a coenzyme to. And the first is pyruvate decarboxylase. And this enzyme, pyruvate decarboxylase, you'll find it in, say, ethanol fermentation. So, generally, I'll just mention, you find it in carbohydrate metabolism, where you see that you have pyruvate, which is undergoing um, some decarboxylation reaction, and at the same time some reduction reaction. Even from the name, pyruvate decarboxylase. Decarboxylase means that removal of carboxyl group. I don't know if on your screen you can see that there's a bond which is red in that molecule. So that's a keto acid. We say a keto acid because it's having a ketone group, C double bond O, and then there is also that carboxylic acid group there. And the bond which is between, these two, carbonyl groups, the keto group and the carboxylic acid group, that is the bond on which the enzyme pyruvate decarboxylase is acting, breaking that bond, so that you have this COO removed as carbon dioxide, and in place of it, hydrogen binds. And so you have a bond which is cleaved, which is shown red there, and you have a bond, a new bond, which is formed, where you now have an aldehyde formed. That is also red. So this is an example of a reaction in which thymine pyrophosphate is involved as a coenzyme to the enzyme pyruvate decarboxylase. Another example you see is with pyruvate dehydrogenase. And also you have the alpha ketoglutarate dehydrogenase. And we did see what dehydrogenases were the last time. Dehydrogenase, that's a synonym for oxidoreductase. When we were looking at the classification of the enzymes, the first class that we saw was the oxidoreductases, which were involved in the oxidation reduction reactions. And here we see pyruvate dehydrogenase is involved in the synthesis of acetyl-CoA. And so, I mean, if it's involved in the formation of acetyl-CoA, it's just like the first reaction which we have seen there. But we see that bond, which is between the keto group and the carboxylic acid group, is broken. That COO is given out as carbon dioxide. And in place of the hydrogen, which you see in that first reaction where you have an aldehyde from, this time around, the coenzyme A binds. So that is where you are having a formation of the acetyl-CoA. And you also see that it says the alpha ketoglutarate dehydrogenase is acting in the citric acid cycle. Of course, this acetyl-CoA goes into the TCA cycle. And then you have the third example here, transketolase. It's involved in carbon assimilation reactions. And it's a pentose phosphate pathway, one of the carbohydrates metabolic pathways, where you have the synthesis of the ribose 5 phosphates, and it is that ribose 5 phosphate which will then go into the synthesis of nucleotides, which goes into the synthesis of nucleic acids, RNA, and DNA. And here too we see there's a bond red, that bond between the keto carbon and the carbon carrying the hydroxyl group. That bond is what is broken, and when it is broken, another group altogether comes to bind to it. The group looks similar, but it's not the same. That's why we have this to be R4 and ask this to be R5. So you have the thymine pyrophosphate involved in the breakdown of bonds between carbonyl groups, such as what we have seen between keto groups and carboxyl groups. Or you have this a keto group and a carbon with a hydroxyl group on it. You find thymine pyrophosphate acting in such reactions. I think we can look at mechanism of action of thymine here, of thymine pyrophosphate, to see how such a reaction occurs. Now, before we start to talk about what really is happening over here, I usually make an analogy of, let's say you are in the kitchen and you want to slice some tomatoes, some vegetables, maybe some cucumber. Usually, you don't hold the whichever vegetable it is that you want to cut. It's more comfortable when you place it on a chopping board, than to be, say it's in the air and you are trying to use your knife to chop it in the air. Even if you don't have a chopping board, you will definitely place it in your palm before you try to chop it. So you give some stability to the vegetable before you chop it into pieces. So why am I making this analogy? The enzyme is your knife that you are using on the substrate, which is the vegetable. And the chopping board on which you have placed the vegetable to give it stability before you chop it becomes the coenzyme. So that is the essence of the analogy that I'm making. So what we'll do now, you see, these reactions over here are labeled from number 1 up to number 5. So there are 5 different reactions that are occurring over here. These things are not things that you should cramp in your head anyway. It's only for you to understand, step-by-step processes of what occurs. Usually when we talk about these chemical reactions, I usually mention to you that a lot of times you see reaction being shown as some reactants, an arrow, and then it gives you a product. Sometimes it can be oversimplified that you think that is also for instance, this whole reaction could just be written as say this is pyruvate. So that is the reactant, the product, and that pyruvate gives you acetaldehyde. That's the reaction we just saw in the table, where you saw pyruvate and the carboxyl group was broken off. And when the carboxyl group was broken off, then you had hydrogen coming to bind onto the carbon there, and then you had acetaldehyde. So it could be pyruvate. There will just be one arrow up to acetaldehyde. And maybe on top of the arrow, you can make it more complex to, have to show that carbon dioxide is giving out, and then acetaldehyde is there. That's just one arrow, and it's telling you this is the reaction. Very simple, like that. But if you do analyze the reaction more critically and look at step-by-step processes through which pyruvate undergoes before it becomes acetaldehyde, that's when you start looking at this mechanism of reactions, which sometimes is intimidating. I understand it, that it can be intimidating to you when you see all the reactions at once. But I always encourage you, students, don't be scared of these things. Just take one arrow at a time and just try to make sense out of what really is happening, arrow by arrow. What is it that is happening? And enjoy to understand whatever is happening, at least one arrow. The next time you look at the next arrow and try to understand what it is which is happening. Now, let's take the first arrow, arrow number 1. And you see, we did mention the fact that when you take thiamine pyrophosphate, there is a particular portion of the molecule which was active. You remember it was the thiazolium ring. And even on the thiazolium ring, there was a particular carbon number 2, on which there was a reactive hydrogen there. You remember that. So we are not showing the full molecule of the thiamine pyrophosphate, but we're just taking that particular portion of the thiamine pyrophosphate, which is the thiazolium ring, and that particular carbon there is what is interacting with the substrate, the pyruvate. And you see the hydrogen is not there, because that was the reactive hydrogen, which easily goes off, and you have a carbon ion. So that carbon is carrying a negative charge, which can have a nucleophilic attack on that carbon with the keto group there. And what you do see is that now pyruvate, which is the substrate, is now bonded to the thiazolium ring. So that is the coenzyme that you now have the substrate sitting on it. The substrate is bonded to it. So you see the analogy I made? You are having the thiamine pyrophosphate as a chopping board, and the substrate, pyruvate, is on it, getting stabilized on it before the enzyme will act on it. And from there, what you do have is you have a carboxylic acid group is being removed. So you see reaction number 2, you see that carbon dioxide is going out. That is the decarboxylation reaction. We did say pyruvate decarboxylase. That's the enzyme that is acting. And thiamine pyrophosphate is acting as a coenzyme. And so this is a function of the coenzyme here now, that the coenzyme, is giving that stability to the substrates for the reaction to occur on it. Decarboxylation, and you have carbon dioxide given out. From there on, you are left with these resonance forms. Either you are having a CH3, COH, and you have the carbon forming a double bond with the coenzyme, or you are having the carbon forming a single bond with the coenzyme, but that carbon is carrying a negative charge. So these are resonance forms, like some isomeric forms of these. These are unstable. And so quickly, you have a third reaction where, so if you have that carbon ion, because the carbon is carrying a negative charge on it, it can easily attract a proton. And so you see in the next reaction, a proton, H+, reaction number 3, is coming to bind onto that carbon there. And the next thing you see is that you have that hydroxyl group, being reduced to an aldehyde group. And even on that carbon, you see that you have a C double bond O. So what happens is you have that COH being oxidized to a C double bond O. And so now you have the acetaldehyde, which is the product which is coming out of that reaction. So that's a dissociation of now the substrate from the coenzyme to have the product acetaldehyde. And so you now have the coenzyme free. And that coenzyme, you see, hydrogen ion binding back onto it and can go back into the reaction, that cycle of interacting with another pyruvate molecule, and the reaction cycle continues like that, on and on and on. So like I said from the beginning, it's not something which you should cramp in your head. It's only for you to understand that there are step-by-step processes that go on when you see reactions with just one arrow. It's a simplification of what is happening. And this whole thing, as I said from the beginning, could be written as pyruvate, one arrow, and then it will be shown that carbon dioxide is given out on that arrow, and the product is acetaldehyde. Finished. But if you take the mechanism of reaction, this is actually what is happening. So this is how to this is just for you to see the activity of thymine pyrophosphate, what thymine pyrophosphate is doing as a coenzyme. We take another coenzyme. This one too, you have been seeing it. You may not pay attention, but today your attention is being drawn to it. Nicotinamide nucleotides. You see in your biochemistry books this abbreviation, NAD, NADP+, NADPH. These are the nicotinamide nucleotides. They also serve as coenzymes. And here you see niacin. We did see niacin as one of the vitamin Bs. So that is niacin, or nicotinic acid. And when it gets aminated, it becomes nicotinamide. So just in place of, if this was written in its molecular form, this will be an OH. So in place of it, you now have NH2. So it's an amide of niacin, an amide of nicotinic acid. That's nicotinamide. And you have seen this nucleotide where you have the ribose with adenine, the nitrogenous base adenine on it, and a phosphate group there on carbon number 6. Or is it carbon number 5? Because that's a pentose on carbon number 5. And you refer to it as adenosine. So that's one nucleotide, adenosine. But if you pay close attention, you realize that there is a second nucleotide where you have a ribose, and the base, which is on the ribose here, is now the nicotinamide. So that's nicotinamide there. And there's a phosphate group on carbon number 5. And you do see that the two phosphate groups are now bonded. So that's a phosphodiester bond. This looks much like the bond which you find between nucleotides in nucleic acid strand, like RNA and DNA. It's a phosphodiester bond, which you are seeing there. And because you have these two nucleotides being bonded this way, you refer to it as a dinucleotide. So that's why you see NAD or NADP or NADPH, because nicotinamide adenine. So it's referring to the bases first, nicotinamide, then adenine. And it says dinucleotide. So two nucleotides bonded together. And if you have because you're having these phosphate groups, you do refer to it as a nicotinamide dinucleotide phosphate. Sometimes you see NADPH. That is if it's in its reduced form. This way, it's in its oxidized form, and so you have it being referred to as a nicotinamide dinucleotide phosphate. So it's a structure. So structure of the nicotinamide nucleotide. You will see in some of the structures, in place of this hydroxyl group, there can be another phosphate group there. So when the phosphate group is there, that's when you see it as NADP plus. But if there's no phosphate group there, it's just NAD. All right, so that's about the structure of the nicotinamide nucleotides. So this is just like what we have been doing. I think I explained it to you on many occasions that in biochemistry, what we do is, if you take a substance, you want to understand the structure of the substance. And then when you finish looking at the structure, you then want to now understand the function of that particular structure that you have just looked at. So it's the same thing we are doing. You can see that when we took thymine pyrophosphate, we first looked at its structure. And then after looking at the structure, we went to look at its function. We are doing the same thing again with this one, nicotinamide adenine dinucleotide. We say nicotinamide nucleotides generally. We look at the structure, and then after that, we start to look at the function. Now, this is nicotinamide nucleotide here, the structure that we have seen already. And here we can talk about another kind of specificity. You know, with the last time we did talk about some stereospecificity, where you had enzymes acting on some specific stereoisomer. Either is an L isomer or a D isomer. We did see these things. And here too, you have this type of specificity where you have, if you look at the structure of the nicotinamide nucleotide, you can either have the front of the nicotinamide base or the back of it that will be having interaction with the substrate, or where you have protons or hydride ions binding to. That's some form of specificity that you come across with these coenzymes. And so you see in that first reaction, you are having because these nicotinamide nucleotides are involved basically in oxidation reduction reactions. They are involved in the transfer of electrons. So they are referred to as electron carriers. And usually these electrons, are carried together with protons, like hydrogen atoms. So you see that's hydrogen there, which is blue, see it comes to bind on the nicotinamide there. And we are referring to this as the A side. That's like the front side of the nicotinamide base. And you see we refer to this as the B side. That is the back. So you see the hydrogen is at the back. The hydrogen that comes to bind is binding to the back of it. So it's a form of specificity depending on the enzyme to which the nicotinamide nucleotide is acting as a coenzyme, you have the proton or the hydrogen ion binding either to the front side or to the back side. We'll see some examples of, say, enzymes with such specificity. And so the function of the nicotinamide nucleotides is what? They are electron carriers. And where will that electron, the electron carriers, where will they be acting? They will be acting in the oxidation reduction reactions, which means that the nicotinamide nucleotides will be coenzymes to which class of enzymes? To the oxidoreductases, or what we also refer to as the dehydrogenases. All right. Now the stereospecificity that I was talking about earlier, we see some examples of enzymes here that you find a nicotinamide nucleotide acting as a coenzyme to. And we see the stereospecificity being referred to over here in the third column. So for isocitrate dehydrogenase, the nicotinamide nucleotide will be binding to the front side, the A side. For alpha-ketoglutarate dehydrogenase, it will be binding to the B side, the back side. The next one to the same, glucose 6-phosphate dehydrogenase, that specificity is that you have NADP plus as the coenzyme, and it will be binding to the back side. And so you see malate dehydrogenase, front side, glutamate dehydrogenase, back side, glyceraldehyde 3-phosphate dehydrogenase, the back side. And the last two, lactate dehydrogenase and alcohol dehydrogenase, you'll find that the hydrogen ion is going to be binding to the front side of the NAD or the nicotinamide nucleotide as the coenzyme. I mean, that is in the process of the transfer of electrons. You find out the hydrogen ion, the proton, which is accompanying the electron which is being transferred, is either binding to the front side or the back side of the nicotinamide base in that nicotinamide nucleotide. So this is another form of stereospecificity that you can add to what we studied the last time. Right. Now we take some specific example of reaction in which you will see this nicotinamide nucleotide acting. This is with the enzyme alcohol dehydrogenase, and you see NAD plus NADH acting in that reaction as a coenzyme to the alcohol dehydrogenase. And here you see ethanol is being oxidized to acetaldehyde. And acetaldehyde, so the reaction continues. Acetaldehyde gets converted to acetic acid. It's a common reaction to do with alcohol metabolism. And the enzyme that is involved in that process is the alcohol dehydrogenase. And the coenzyme there is a nicotinamide dinucleotide that is involved. All right. Now, we take another type of nucleotide, what we refer to as the flavin nucleotide. The flavin nucleotide. So the here too is a dinucleotide. You see it being written as FAD. FAD, FADH, FADH2. You'll be seeing those things. And this is how the structure looks like. So you have this coenzymes coming from vitamin B2. That is riboflavin. So you see the name, riboflavin, because it's coming from this flavin. Now, here too, you see there is nicotinamide. No, not nicotinamide per se. That is adenine. This adenine that is binding on the ribose sugar. It's a phosphate group on it. So that is one nucleotide. See the dashed lines I've put there to show that that's the end of this nucleotide. So that is, say, adenosine. You have adenine on the sugar with a phosphate. So that's one nucleotide. Now this nucleotide, which is the flavin mononucleotide, is written, the sugar is written as a straight chain. But you shouldn't have a problem with this because we did look at the carbohydrate structures in first year, where we saw the straight chain structures. And so you shouldn't have a problem with that. If you look at it, this is 1, 2, 3, 4, 5 carbons there. So that is also a pentose with a phosphate group on it. And then you have that flavin, what we refer to as the isoalloxacin ring. So that is also like a nitrogenous base. A nitrogenous base. So it's that in place of these bases which you have been seeing before, you now have that isoalloxacin ring. And because there's only one phosphate group there, you have that as a mononucleotide. FMN only means flavin mononucleotide. But if you having two nucleotides that are bonded together, just like what we saw with the nicotinamide nucleotides, then you have a flavin adenine dinucleotide, which you see most often, being written with this abbreviation, FAD. And here too, what do you see happening? Similar thing where you are having transfer of electrons. And so you can have here too the hydride, binding, or let's say only one hydride binding, where you have what we refer to as a semiquinone. That is to say that the nucleotide is not fully protonated. It's only one hydrogen that it takes. If it takes a second hydrogen, you see now it's being written as FADH2. But if it's only one, it's FADH. Sometimes it's shown with a dot on it, which means it's semi-reduced, but here it is fully reduced. So these are also involved in the transfer of electrons, but these usually, they actually bind to the protein. You see, if you look at the nicotinamide nucleotides, what we saw much earlier, we said they are water-soluble coenzymes. They actually move. They move from one molecule to the other. But if you come to these the flavin nucleotides, they actually bind to these proteins, what you do refer to as flavoproteins. So you find a lot of these enzymes in the liver, which are flavoproteins. You have the flavin nucleotide actually bound, tightly bound to these proteins, and they are involved in the transfer of electrons. And here too, so as we did with the nicotinamide nucleotides, you see that there are some examples of enzymes like the acyl-CoA dehydrogenase, which is found in lipid metabolism. FAD is a coenzyme. dihydrolipoldehydrogenase. This one too, lipid metabolism, FAD. Succinate dehydrogenase, that's in carbohydrate metabolism. You find the FAD as a coenzyme, glycerol 3-phosphate dehydrogenase. That's you find also in carbohydrate metabolism, FAD there. Thioredoxin reductase, that you find in the protein metabolism, FAD. You have the NADH dehydrogenase complex 1. That too you'll find in different metabolic reactions. Glycolate oxidase you find in carbohydrate metabolism. Both of them, you see the coenzyme there is not a dinucleotide, but it's a flavin mononucleotide, FMN. So these are some of the functions of this coenzyme. Now these flavin nucleotides, I did mention that they are tightly bound to the enzymes and that they are acting as coenzymes too, and so we refer to these enzymes as flavoproteins. And you'll find these flavoproteins acting as what we refer to as light receptors in what we also call cryptochromes. They mediate effect of blue light in plants. If you do observe that in the morning, for instance, there are plants, you see that the leaves are opening, you have the flowers opening, and in the evenings, the flowers close or the leaves close. That's a form of what we refer to as a biological clock, what we refer to as a circadian rhythm in mammals. So for human beings, for instance, we do have this circadian rhythm that when it is in the night, when it gets dark, we should feel sleepy and go to bed. And in the morning, when it gets bright, the light should activate us to get up, to wake up. I mean, that's a normal rhythm, what we refer to as a biological clock or the circadian rhythm. A lot of these flavoproteins act in this process of the circadian rhythm because they react to light. So you have some of these proteins in the eyes that react to this rhythm of light over different times of the day. Now you do have some of these also acting in say repair of nucleic acids when there is some defect in the nucleic acids. You have a repair of this. And these are what you do refer to as the photolysis. You do remember the one class of the enzymes that we refer to were lyases, involved in either removal of groups to form double bonds or the addition of groups to remove double bonds. And the ones that react with, they actually react to light. You find these in prokaryotes and even some eukaryotes, which are involved in the repair of defects in nucleic acids like DNA. Find these flavoproteins active in those photolysis. That class of enzymes, photolysis. You find the flavin nucleotide as a coenzyme for those enzymes. Now we take a look at the structure of coenzyme A. Yeah, what you also see is one of these common coenzymes that is referred to a lot of times in biochemistry. I'm sure by now you would have heard of it, you would have seen it when you were looking at metabolism of carbohydrates. You by all means came across acetyl-CoA. And so usually you just write it in a short form like that. Sometimes it's just written like in words like acetyl-CoA. In the structure, the acetyl group, which is blue, is shown, and then is bonded to the coenzyme A, which is also shortened in that way. So that coenzyme A, which you are seeing written that in that very short form, S-CoA, is the structure that you are seeing. So you can see it's such a large structure. So now you understand why a lot of times it's just being written coenzyme A or this S-CoA, because of course you can't be writing all this all the time. And here too, you see that you have a phosphoadenosine. So that's another nucleotide. It's a phosphonucleotide there. On carbon number 3, you have a phosphate group. So that's why we say it's a phosphoadenosine diphosphate. So 2 phosphate groups there. We did see pantothenic acid. So that's the shaded portion, the pink shaded portion. We did see pantothenic acid as one of the vitamin Bs. And I mentioned that it's just a part of the coenzyme, but itself is not fully a coenzyme. So this is where you are seeing it. So you can take it as the pantothenic acid as a vitamin B in its active form is what you are seeing as the coenzyme A, because now it's bonded onto a phospho, diphosphonucleotide, and also to another, some derivative of amino acid, the beta-mercaptoethylamine. But you see, all these big structure which you are seeing over here, the reactive portion of it is what is in red you are seeing there. The sulfur hydral group at the end there is being shown as the reactive thiol group. It is at that particular end of the molecule that you have interaction with a substrate. So you see it clearly being depicted here in the acetyl-CoA, where you see that, that S red is what you see there, the sulfur, and the CoA is just, referring to the rest of the structure, the big structure there. So it is that particular portion. So when you have that hydrogen now given out, you can have the sulfur binding on the acetyl group to give you the acetyl-CoA. So, I mean, even if you don't understand anything at all, you should remember that the reactive portion of the coenzyme A is this thiol group or the sulfur hydral group at the end of the molecule. Biotin. Biotin is another vitamin B that we saw, which is also involved in the transfer of carboxyl groups. We did see it in the table, but here we are looking at a reaction to see how biotin actually carries out its function. And you see here, this is a bicarbonate ion having this carboxyl group, which is in red. And you see pyruvate. We have seen the structure already. Pyruvate, a keto acid. And we see the enzyme, pyruvate carboxylase. So this time around is a carboxylation reaction, not decarboxylation, but carboxylation. That's an opposite. It's an addition of a carboxyl group. It's a transfer of a carboxyl group from bicarbonate onto pyruvate to give you the product oxaloacetate. And so you see, in the end here of pyruvate molecule where you see CH3, in place of one of the hydrogens there, because now you now have a CH2, you have this group, the carboxyl group coming from the bicarbonate there to give you oxaloacetate. This is a common reaction also which you see in the Krebs cycle. I think you remember the Krebs cycle, the very first reaction there was that you have pyruvate coming in that you have acetyl-CoA, which is coming into that reaction, and that's pyruvate, binds onto one of the intermediates and forms oxaloacetate. So I think oxaloacetate then goes to say goes through some decarboxylation reaction for you to get a citrate. You can just go take a look at that the TCA cycle and try to make some sense out of it, and you will understand what it is. So I think this is an opposite of that first reaction, which is in the TCA cycle. And here you only see that there is some expenditure of energy in the form of ATP, and biotin is a coenzyme. It is being shown in that reaction there. So yes, biotin, you see, that's a structure you see on the left-hand side, biotin, and it is involved in the transfer of carboxyl group, the COO group, from one molecule onto the other, and that is a carboxylation reaction. You can see it's a mechanism of action of biotin much more closely, so that if this is the structure which we have just seen, biotin, and it is bonded onto what we are referring to as a biotin carrier protein, take that to be the enzyme itself. So that's an enzyme to which the coenzyme biotin is bonded. And that coenzyme biotin is carrying carboxyl group, which is shaded green, from the bicarbonate ion. And that next reaction, you have the enzyme transcarboxylase, which then transfers that carboxyl group that biotin is carrying onto acetyl-CoA to give you malonyl-CoA. And malonyl, I think this reaction to you come across in lipid metabolism, the conversion of acetyl-CoA to malonyl-CoA, or even the opposite of it, where you have usually when you have this odd number of carbons in a fatty acid, and the fatty acids are being broken down by the process of what we refer to as a beta oxidation. The very last fragment which is left, which is going to be having an odd number of carbons, which is 3 carbons, and that will have some carboxylation onto it, and before you then will be having 2 molecules of acetyl-CoA coming from it. All right, so you will see the mechanism of biotin in say metabolism of lipids. Now I think this should be the last one, folate, what we do refer to as the folic acid. I mean, you just look at the structure. You see that it's having glutamate. That's an amino acid, so glutamic acid. And then you have a middle portion, which is the para-aminobenzoate or the para-aminobenzoic acid. And then you have a methylpterine ring. That's a fused, 6-sided rings. But here too, it's a big molecule, but we have these two nitrogens, N5 and N10, which are blue. And the reason I made that blue is to show that these are the particular portions that are reactive. These are the particular portions that are active to be interacting with the substrate. So you have that, the this is a tetrahydrofolate. But I mean, generally, this is a general structure for folates or folic acids. And what do these do? The function. I think some of you can quickly remember and know that usually folic acid is prescribed for women in pregnancy. What's the reason? That's because the folic acid is involved in several what we say proliferation or specialization of cells. During the development of cells, folic acid is involved as a coenzyme in many biochemical processes, and particularly for the formation of what we refer to as the neural tubes, or let's say the brain of the fetus which is developing. And so when there is a deficiency of folic acid in pregnant women, that can result into some mental retardation, that can result into some growth abnormalities, deformities, and so which is why a lot of times it's prescribed for pregnant women to prevent such, particularly the risk of developing defects in the development of the brain. Because, at the point of the development of the fetus, the process of cell division that is occurring is very fast and very vigorous. And so a lot of folic acid is needed in these processes. And folic acid is involved in reactions of, transfer of what we refer to as single carbon groups. It could be methyl group, methylene group, formyl groups. So these are, carbon groups that are involved in various, biosynthesis. That is, the build-up of different biomolecules, proteins, carbohydrates, nucleic acids. You find folic acid playing very vital roles in those processes. All right, I think this is where I'll keep quiet and listen to you. If you have any questions, you have any contributions, the chat is active. You can write in the chat. I will respond to it. Alternatively, you can raise your hand and I will enable your microphone, call your name, and then you can speak. Then I respond. The floor is open.